Welcome back, everyone. We're going to continue on with our next experiment this week. This week is the ligation and bacterial transformation experiment. And you guys can follow along with me in your lab manuals on page 21. So to begin, I want to talk about ligation, and to do that, we need to talk about the enzymes known as ligases. So what ligases do, they basically just join fragments of DNA back together by stitching up the DNA backbone. So the only condition that needs to be met is these DNA fragments must have matching complementary ends in order for them to be joined back together. So the way that the ligases end up working is, remember that DNA, the nucleotides at least, are able to bind to each other naturally through hydrogen bonding. What the ligases do are actually fix the little nicks or little gaps in the phosphate backbone of our DNA. And the way they do this is by catalyzing the reaction between the phosphate on the 5' end of one fragment to the hydroxyl group sticking off the 3' end of the other. So, of course, this reaction is done many times in the cell, primarily during DNA replication, but it can also be used to kind of help the cell in when the DNA gets damaged by some kind of environmental factors, such as UV light. In fact, damages to our DNA can happen many times. Up to 1,000,000 breaks can occur in a single human cell, which is what makes ligation so important in the cell. So here I kind of want to give a little, diagram showing what I mean. So here we have our complementary ends that will be able to match up together. And when they align, the nucleotides are able to bond via hydrogen bonding. But what's left are these little gaps that you can kind of see here. And this is what the ligase fix. It'll take the phosphate on the 5' end of one DNA and attach it to the hydroxyl end of the 3' end, and vice versa here. And once this reaction once this reaction is done, we'll be able to see that the backbone is completely fixed. So how can we do this inside of the lab? How can we perform a ligation in vitro? So what we want to do is we need to make sure that we have all the components for a ligation. And that means we need to have the complementary DNA fragments, we need to have a ligase, we need to have a buffer that allows for our ligase to work very well, and we need an energy source, meaning for a ligation to occur, we need to have ATP. So remember last week how we prepared both a plasmid as well as our beta-gal gene by digesting it with restriction enzymes? So the reason that we treated them with these restriction enzymes is so that we can actually ligate them together. Both the plasmid as well as our gene were both digested with the same restriction enzymes. So they should be able to match up together and we'll be able to attach our beta-gal gene into the plasmid. So you can see why ligation is so useful, especially in DNA cloning. So another example of how ligase works. So you can see that here we have our complementary ends that match up together, but we still have a little bit of a gap. The ligase is able to come and just seal the phosphate gap. When we look at this in a plasmid, we're able to see exactly what I mean. So remember that, let's look at this end. So on this end, we can see that our plasmid, so this side, was cut with BAM-H1, and our gene on this side was also cut with BAM-H1. That's what lets these two, complementary ends match up together. And the same can be said on the other side with HIND3. So because they're able to link up back together and then the ligase will come and fix this plasmid, once this plasmid is whole, we call it a recombinant plasmid. So there are a couple ways that a ligation can occur, wrong. So we want to make sure that we put in our gene in a very proper direction. And that's kind of why we use the two restriction enzymes that cut in these sticky ends. So let's say we had cut with blunt ends. So instead of cutting jagged, they cut straight through. Then there's a very good chance that when we put in our gene with blunt ends, that it can go in either way. There's a 50-50 chance it'll go counterclockwise or the actual direction, which is clockwise, which is not what we want. We want it to go in the proper direction. We also made sure to use two restriction enzymes instead of just one, because if we had only used one to kind of cut open this plasmid, when we do the ligation reaction, then this can match up again. By using two, we can guarantee that our HIND3 will never match up with the BAM restriction site. So these should never come back together and close up on itself. Same thing by making sure we add the proper recognition sites to the beginning of our primers. We're able to force it so that that our BAM site matches the BAM site on the plasmid. So there was a lot of things that we did to kind of manipulate our gene and our plasmid so that it goes in the right way. Another thing that we're going to do for our ligation reaction to make sure that the reaction happens naturally and we're able to get as much product as possible is we're going to have a 1:3 ratio of vector to gene. So this is because when we mix these two together, we're kind of just giving them as much time as possible for this to occur, because what's really going on is this gene is just floating around in solution, and we're really just hoping that the ends will find each other. And so because we're kind of just relying on luck, we want to make sure we have a high ratio of gene in there just to kind of push the probability closer to them meeting up. We can, of course, go higher than this, but some research has shown that going too high can cause some weird plasmids where we have giant plasmids where like two plasmids will come together with two genes and it'll be really funky. So really 1:3 is probably the best ratio for us. So that's barely the first part of the experiment, just the ligation. So that's just making our recombinant plasmid. The actual transformation is taking this recombinant plasmid and introducing it into our bacterial cells. So for the transformation, we're going to use a special type of bacterial cell known as competent cells. So the cell line that we're using are E. coli BL21 DE3 cells. So this is the cell line. What competent cells mean is that this cell has been manipulated in some way so that they have an easier time taking in foreign DNA. So they're really good to have, they undergo transformation very well. So what you need to know in terms of just our bacterial cells is that the cell is E. coli and the strain BL21, what that means is that it basically is just made to express protein very, very well. And the DE3 is what that's supposed to tell you is that the somewhere in these E. coli cells is a gene that will encode for the T7 DNA polymerase, which will be important a little bit later on when we talk about expression. But that's what you need to know about our cell lines, because that is actually very important for this to work. So that's just our bacterial cells. So how do we make it competent? So depending on the lab, you can make it competent through electroporation, through pulsating it. But for us, we do we use like a chemical treatment to treat our cells. And that is basically treating them with divalent cations. For us specifically, that's calcium. So when we add these divalent cations, they're able to kind of attach themselves very weakly to the membrane. I've got to kind of emphasize that it's a very weak interaction, but they just attach themselves to the membrane and just sit there. And why is this important that we add these divalent cations? it's because of this next step where we heat shock our cells. So we'll take our cells that have been treated with these cations, where we have also added our plasmid, and what we're going to do is just put them into a hot water bath at around 42 degrees and just kind of shock them with some heat. The reason that we want to do this is that the heat will actually cause pores to open up in the membrane, which will allow for our plasmid to kind of go in. What these divalent cations do is, because they're positively charged and kind of sticking to the membrane, they attract the negatively charged plasmid close to the membrane, so that when the pores actually open up, there's a high likelihood that they'll actually go inside of the cell. We just have to be careful when we heat shock a cell, because of course if we heat shock it too long, then the cell can actually get damaged to the point where it'll just fall apart because the pores get too big, things will leak out of the cell, and the membrane will just end up breaking. On the other side, if we don't do it for enough time, then the pores don't actually open up enough for the plasmid to go in, and we get absolutely no transformation. So there's actually just a very narrow window for this heat shock to work properly. Another thing I kind of want to emphasize is the use of an antibiotic resistance gene when we use our plasmids. So here I kind of just want to skip to a nice little figure. So this is kind of a little figure of our plasmid, our PET30A plasmid that we have inserted our gene into. And what you can see is that there's three key, components to this plasmid. The first is a promoter that's going to drive the transcription of our gene. The second is our actual target gene, which is the beta-gal gene. And finally, somewhere in the plasmid, specifically in the PET30A plasmid, is an antibiotic resistance gene. And for PET30, that antibiotic is ampicillin. So why do we need to have an antibiotic inside the plasmid? It's for a later step when we want to check our cells to see if they actually took in the plasmid with the gene. So once we heat shock our cells and they take in the plasmid, we just grow them up in some media and then eventually plate them in an agar plate. This agar plate will actually have some kind of ampicillin added into it, so that when we plate these cells, only the cells with the antibiotic resistance gene will be able to grow. So what this means is that only cells that took in the plasmid will be able to grow. Does this mean that all of the cells on the plate will have our gene? No, it does not. There could be some rare occurrence where maybe the plasmid didn't get digested properly and it continued through the entire process, and we just continued with this one circular piece of plasmid that never got digested but got taken in by a cell, and so it's going to grow on the plate. So that can occur. However, the likelihood is very, very low. But it's always useful to just check just to be 100% certain. So once a plate grows, what we end up doing is we actually check a couple of the colonies using PCR to see if the gene is still there. So similar to the very first PCR we did in the lab, all we do is we set up everything the exact same way, but instead of using a beta-gal gene as a template, we use our actual colony as the template. So we kind of just poke it and mix it in with our PCR reaction and just let it go. Once we get the results, we should be able to see if there is a band that's very, indicative of there being a beta-gal gene somewhere in that colony. Or there's a beta-gal gene in that colony because it actually grew. And if we see no band, then that's another good indication that the colony didn't have our gene and it was just the plasmid. But that's pretty much everything for the whole ligation and transformation. And once we are done with this next step, once we're done with this experiment, the next step we'll be doing is actually growing up the colonies so that we can try to express our beta-gal protein.